Hello, biology students, and welcome to Chapter 15, which is going to cover the senses. Now, the first thing before we get into this chapter I want to talk about is what are our senses? Most of you guys have learned that our senses are, um, based on 5 things. It's either touch, sight, taste, smell, and hearing, or one of these has been replaced with equilibrium. But the truth is, these days, everybody is kind of taking it and making them more specific. For instance, touch can be broken down into itching is a sense, pressure is a sense. But the true definition of what a sense is going to be any system that consists of a group of sensory cell types that respond to a specific physical phenomenon, and that corresponds to a particular group of regions within our brain where the signals are going to be received and interpreted. So like I said, this could be broken down into so many different categories. Another one would be proprioception, which is if you close your eyes and take your finger and touch it to your nose, that's proprioception. It's knowing where your body is in space. So anyway, let's dive in. We've kind of grouped them together. So we'll talk about each of the senses, more specifically sight and hearing in this lecture. And we'll dive more into this within your anatomy courses. So our points to ponder. What are sensory receptors? How do we detect the sense of taste and smell? What is the anatomy of the eye? How do we focus images? What are some eye abnormalities? What is the anatomy of the ear? Which parts function in balance and which parts function in hearing? So let's talk about our receptors first. Remember, a receptor can lie within any organ within our body, and it's going to sense something. It's going to pull information and usually send it over to our central nervous system, which always consists of our spinal cord and brain. So, sensory receptors. They're dendrites specialized to detect certain types of stimuli. Now, what is a dendrite? Let's talk about that first. A dendrite is a part of a neuron. A neuron is the same thing as a nerve. So our nerves, they have these spiky extensions. Okay, here's the cell body, because a nerve is a cell. There's my axon. This is the axon terminal. Okay, so information, switch our color here. Information gets picked up by these spiky extensions called dendrites. And then it transmits this information to the axon, moves through the axon toward the end of the neuron, and then will synapse or send this impulse to either another nerve or another organ. So we have specific receptors for every type of stimuli that we can sense. And we divide these into enteroceptors, which are going to detect stimuli from outside the body, like tasting something salty, hearing the ambulance, or seeing a pretty flower, okay? And then our interoceptors, they're going to receive stimuli from the inside of the body. So a change in our blood pressure. So for example, we have something called baroreceptors in our aortic arch and carotid sinus here that'll send information to our brain telling us, oh, your blood pressure is lowered, okay? And it'll do the proper changes in function to bring that blood pressure up. Or for instance, we have receptors in the stomach that will give us the sense of maybe our stomach is stretching too much and we feel really full, or something is bothering us and our stomach is going to give us a sense of pain. Those are interoceptors. So we're just going to talk about some specific sensory receptors here. We have chemoreceptors that'll respond to nearby chemicals. So these are going to be in areas actually in our nose. So when we take in a sense of smell, let's say we're smelling a rose, the smell of a rose are actually broken down into a bunch of different chemicals and are picked up within our nose by these chemoreceptors within the mucosa. We also have nociceptors, which are pain receptors, and these are going to be our chemoreceptors that respond to chemicals released by damaged tissue. I also forgot to mention that we have our carotid sinus here. This is an example of a chemoreceptor where we would have the ability to pick up the sense of a change in blood pressure. So nociceptor, like I said, that's our pain receptor. So these you can find in our skin, okay? Where if you would have inflammation or cut somewhere, they would be triggered. Okay, now on to photoreceptors. These are going to be located within the retina of our eye. So you see these photoreceptors called rods and cones, which we'll talk about in a moment, all the way in our retina, all the way toward the back. And they pick up light energy and will transmit this to our optic nerve, which will go and eventually get to our occipital lobe to give us a sensory information on what we're seeing in front of us. Our mechanoreceptors, most of these can be found in the skin as well. They're going to respond to mechanical forces such as pressure. So we have a couple different types of mechanoreceptors for pressure. For example, we have our Pacinian corpuscles here that are going to sense deep, strong pressure. We also have some Meissner's corpuscles that are going to lie here in between the epidermis and the dermis that'll sense light touch. More of these we've covered in our integumentary system, so I won't go through all of them here. And lastly, we have our thermoreceptors. Thermo referring to temperature. These are going to be within our skin as well and will tell us if we're sensing things are too hot or too cold. So if you were to put your hand on a hot stove, these thermoreceptors would be triggered and we would have a reflex to pull our hand away because it's too hot, we're going to burn our skin. And I have a breakdown here for you from your textbook, table 15.1, that will tell you some of the sensory receptors we have throughout the body and what the stimulus is broken down to. So like we said, our sense of smell are picked up by olfactory cells, which are really, we have our chemoreceptors in there, and our stimulus is the chemicals that we get from the rose. That's going to help us with the sense of smell, and it's within our olfactory epithelium. Olfactory will always refer to our nose and sense of smell. So that's just one example. I'll let you read through this list on your own. We are going to talk about some of these sensory receptors throughout our lecture, so I won't go through them now. We'll talk about them individually. So how does sensation occur? Well, we already mentioned that our sensory receptors are going to pick up information and send it along a nerve impulse to our central nervous system. So this is a nice little schematic breaking down our nervous systems. So first, what I want you to know before we dive into this is that our CNS, okay, let me circle central nervous system. Our CNS is broken down into the brain and spinal cord. Yes, this is my brain. Wonderful artist here. Everything else is going to fall into our peripheral nervous system. Okay, so these are the nerves that branch out to the sides here. Okay, we also have some cranial nerves that branch out. So just to reiterate, central nervous system consists of the brain and spinal cord. Peripheral nervous system consists of everything else. So our spinal nerves and our cranial nerves. Okay. All right, now from there. So we get the stimulus. Let's say it's within our skin. Let's say that example where we're touching a hot stove. Our thermoreceptors tell us that it's too hot, we're going to burn our hand. That gets sent through a nerve, a sensory receptor, so it'll go through our peripheral nervous system, eventually going to our spinal cord, and sometimes we'll go over to the brain. In our instance, since it's a reflex, we'll just do a quick loop here in our spinal cord to pull our hand back. But let's just say we go up to the brain, our brain is going to interpret that information, and then what happens is we go out another nerve over to, let's say, a muscle. So we want to stimulate our biceps brachii to contract and pull our hand away. Okay, so that's just an example of how sensation occurs. Now sometimes we can have something called sensation adaptation happen. This is when we get a decrease in the stimulus response when we have repetitive stimuli. So for example, odor. So you walk into a classroom and one of your classmates has a lot of perfume on. After 5 to 10 minutes, you're not going to smell it as strongly as you did when you first walked into the room. So your body adapts it. It kind of edits the information that's coming into your body. Another example is the clothes that you're wearing. When you first put it on, you might have more of a consciousness of how that sweater feels on you. But then over time, you almost forget that information. You're not taking it in. Oh, I have my sweater on over and over again. All right. I mentioned proprioception in the beginning of this lecture, and in order to sense proprioception, you need proprioceptors, and these are fit within the subclass mechanoreceptors, and they help us with our reflexes. So most of you already know that knee-jerk reflex, when we hit the patellar tendon with a hammer, it's going to cause us to extend the knee, right? But this is happening all the time throughout our body. It's helping to maintain our muscle tone, meaning our muscles are in partial contraction so that when we need to contract them, it'll happen much faster than if they were completely at rest. And they also help us with our balance or equilibrium and our posture. All right, we already mentioned some of these cutaneous receptors in our integumentary system, and I told you about some of them when we discussed mechanoreceptors. So now we're going to dive into a little bit more detail. Like I said, most of these receptors are located in the skin in the area of the dermis. So let's just review the layers of skin here real quick. We have the epidermis, which is the top portion of the skin. It makes up about 20% of our skin. The other 80% is the dermis. And below that, we have our hypodermis or subcutaneous tissue that's mostly made up of fat. So we have our free nerve endings within the skin. Most of these are going to be nociceptors or pain receptors, and they are also going to consist of those thermoreceptors that are going to sense for temperature, so if something is hot or cold. Below this epidermis, we have our Merkel discs. These are actually connected to cells that lie in the very last layer of our epidermis and will give us a sense of light pressure or light touch. We have our Krauss end bulbs. This is going to help us with our sense of touch as well. We also have nerves that'll go and connect to the root of our hair, and that'll help us with the sense of touch. You know, if you have like a little feather just lightly grazing the hairs, that'll go on down and give us the sense of touch all the way down to the base of the hair, which is the root. Our Ruffini endings are going to assist us with pressure as well. These are located pretty close to the epidermis, but they're in the dermis. And then deep, deep, deep in the dermis, this is actually quite high up, but usually they're down in here, very close to our subcutaneous tissue, will be the Pacinian corpuscles, which will sense for deep pressure. If you look at this on a histology slide, usually it looks like the shape of an onion. You can kind of see how it has those concentric rings. And lastly, we have the Meissner's corpuscles, and these are located at the very top portion of the dermis below the epidermis, and these sense for light pressure as well. Okay, so now we're going to focus on taste, and we have 4,000 taste buds, and these are located on the tongue, and we have 5 main modalities when it comes to taste. We have sweet, sour, salty, bitter, and umami, which is a savory or meaty taste. Most of us are familiar with the other 4, but umami is one of the newer ones. So on the tongue, we actually have 4 different papillae, and these are examples of what the papillae look like. Sometimes you can see them a little bit enlarged when you look at a person's tongue, and along the papillae, so down these sides, that's where our taste receptors are found, or taste buds. So you can see one pictured here in this EM. And this is a zoomed in picture of what a taste bud looks like. We have a sensory nerve that'll be attached to each of these taste cells that will bring in chemicals through the taste pore. Okay, so we bring it in and then we transmit what we're tasting through a sensory nerve, which will go to our brain. We have a specific area in the brain which will interpret the taste that we are sensing. And 80 to 90% of what we perceive as taste is actually due to the sense of smell. And so this is why we can't really taste too well when we have a cold. Our smell receptors are going to depend on 10 to 20 million olfactory cells. Remember when, whoops, sorry about that. When you see this word olfactory, always think sense of smell having to do with the nose. And these are modified neurons. So within our skull, we have this special cranial nerve that comes off of the brain. And then within one of our cranial bones, we have these holes, and these holes provide an exit for the olfactory nerve fibers to move on down into the nasal mucosa and pick up the chemical information from our chemoreceptors here in the nose, from the olfactory cells, and then we'll transmit it along the olfactory nerve and to our specific area of the brain that is created for smell. Oh, and my slide is not advancing for some reason. Let's try this. Oh, now it went too far. Sorry about that. Okay, next we're going to talk about the anatomy of the eye. So our eye can be broken down into 3 different layers. The outermost layer is the sclera. And it's the white part that you can see of your eye, or the very anterior part of your eye where we have our iris, which is the colored part, and the pupil, which is the black circle. So this is very fibrous, and it consists of the cornea. So all of this in white is the sclera, but that part where you see the color of your eye and the pupil, the black part, we see that because this anterior portion is called the cornea. And so it's kind of a window to the eye where we'll be able to see these structures here. Well, really this structure, okay? Our second layer, or middle layer, is going to be the choroid. This red, it's actually more brown in histology slides and everything, pigmented layer here. And this is that dark brown color because, or dark red color, because it's very vascular. There's lots of blood vessels in this area. And lastly, we have the retina, which is pictured in the yellow here. And this is the area where we have our photoreceptors, those rods and cones that I mentioned in the beginning of the lecture that'll help us pick up the sense of sight. So we get light rays being passed to these photoreceptors. We can also break down the eye into 2 different compartments. We have the anterior compartment, which I have in purple there. The anterior compartment is made up of the anterior chamber and the posterior chamber, but all of this anterior compartment is known as the aqueous humor. So I've outlined it in green for you here. And usually this is more of a clear watery fluid, and it's all the area between the cornea, remember the window to our eye, and the lens, which would be this right in here, this oval-shaped lens. Our posterior compartment is filled with a gelatinous type of material, and that is called our vitreous humor, okay? Oh, I just realized that this is not, your posterior compartment is not your posterior chamber, so let me just delete this for you. Let's go back. Okay, so our vitreous humor is basically what our posterior compartment is. It's everything behind this lens. And like I said, it's a gel-like material in here. And this is the diagram that I pulled from your book. Shows you the different layers of the eye. And then we're going to be talking about all of these structures in the coming slides, so I won't go through it now. Okay, so let's start off at our first outermost layer, which is the sclera. Like I said, that's all of this, this white portion of the eye that you can see if you look at yourself or your neighbor. And we have little blood vessels going through them as well. That cornea is the anterior compartment that is transparent. So like we said, the window of the eye where we can see our iris and pupil. And we have our pupil, which is the hole that allows the light to enter into the eye. I forgot to mention as well, our cornea has this bend to it so that it can help to refract the light as it enters in to be displayed on our retina. So we're going to be talking about some refractory light rays in the coming slides as well. So just keep that in mind, that our cornea is going to help to bend the light. You don't see much bending here, but it goes through our pupil and then will go to the retina. All right, our middle layer, the choroid. This is going to absorb light rays, not absorbed by the retina. And it is going to consist of, let me fast forward here a little bit. Where's our iris? There it is. So here is our iris, and keep in mind this is a, basically the eye is cut in half. This is a sagittal cut. So this iris is a continuous circle all the way around. And it has 2 layers of muscles as well. So we can regulate the size of our pupil based on if our muscles relax, that'll cause our pupils to dilate. And if these muscles contract, it'll cause a constricting of the pupil. Our ciliary body contains within it a ciliary muscle, and it's posterior to the iris. And this helps to control the shape of our lens by these suspensory ligaments here. So when it contracts or relaxes, it can pull and narrow the lens, or it can relax and cause the lens to become more circular and wider. And the function of our lens is going to be to refract and focus the light rays onto our retina. So let's talk a little bit more about the lens. This is another view here. So here is our iris. The middle portion where we see this gap is our pupil. Lens is located behind. Here's our suspensory ligaments. These are also called zonule or zonulus ligaments. And then we have our ciliary body with that ciliary muscle within it. So our lens is very flexible, it is transparent, and it's a concave structure. This is going to help us for visual accommodation, and this will happen when the lens changes shape in order to focus the light on the retina and form an image. So we're looking at the letter R here. All of this information goes into the lens, and anything that we have within our view, it's always going to be inverted and backwards, okay? It's just the way the light rays come in and the way our lens, cornea, all of that works together to invert and reverse this image. And as we age, this lens starts to lose elasticity. So we use eyeglasses or contacts to correct this. And this is just another view here. First, let me talk about our sympathetic activation. We're going to have our ciliary muscles relaxed, and this tightens our ligaments, causing the lens to stretch and become more narrow and flattening it. And our parasympathetic activation is going to cause the muscle to contract here, and that loosens these ligaments, hence they have more of that curly look to them, and that will make them make the lens more wide. Tends to make it bulge a little bit more. And then our last layer is the retina. Again, the retina is where we're going to find our photoreceptors, and these are called rods and cones. And you could see the shape of these. These rods are kind of straight looking, and the cones have a cone-shaped head. Our rods are going to make us sensitive to light. So this is more of the black and white color that we pick up, whereas the cones are required for us to see bright light and will respond to wavelengths of color, wavelengths of light. So I always think cone has a C, color has a C, so cones are always for color. Now, our retina has a special area called the fovea centralis. You see how this dips down a little bit here? And this is where we have densely packed amounts of cones where our images are going to focus. So this is the area where we perceive color. Now within the an area called the macula lutea, that's where we have this fovea centralis, just so you're aware. And I think I have another view of this. When you go to the eye doctor, they look at your retina. Sometimes they dilate your eye, and these days they have technology. I think it costs more to do, but they can take pictures of your retina by you looking into this machine. And so you can see an area where we have our optic disc. This is where our optic nerve is going to exit our eye. We'll talk about that in a moment. And the other thing you can see is what we just talked about, this macula lutea, and right in the center is the fovea centralis, where all those cones are found. The rest of this area, as we're moving on away from that fovea centralis, we start to have more and more rods instead of cones. So let's talk about that optic disc in the last image. It is the area where our optic nerve is going to exit. What's our optic nerve? It is a nerve that comes off of our brain. This is known as cranial nerve number 2, and it takes information from our sensory receptors in the eye, which would be our photoreceptors, like the rods and cones, and transmits it along the optic nerve to go eventually over to our occipital lobe in our brain and interpret what we're seeing. Now, this also creates a blind spot for us. And basically it means we don't have any rods or cones in this area. So are we going to be able to see anything? No, we're not. So I had this little activity for you. It's probably not going to work by you looking at the screen. So if we have some time next week, we will try this, but maybe step a little bit further away from the screen, and you're supposed to cover, I believe it is your right eye, and then you could do either one of these. You're going to start looking at number 1, 2, 3, and then by the time you get to the number 4, you're going to start to see this smiley face or this gap disappear, and that's illustrating for you your blind spot. So like I said, it might not work for some of you. It all has to do with the distance you are away from the screen. So maybe you can try it a couple different times and see if it happens for you. And then I believe it's around the number 6 or 7 where if you keep counting down this way, you'll see it reappear again. So let's talk a little bit more about your photoreceptors of the retina. Your rods are going to contain a visual pigment called rhodopsin. So here's our rhodopsin molecule here. And it's going to pick up our light rays and then cause all these reactions to happen in order to send the information to your brain. Like I mentioned, our rods really pick up our sense of black and white. So it's really important for our peripheral vision and also night vision. And we need good vitamin A for proper functioning. So you can get these from like carrots and sweet potatoes. Those all have really good vitamin A. If you want to get it naturally, of course, you can use a supplement as well. And then our cones, they're going to be mostly located in the fovea centralis. As we move away from that fovea centralis, we have less and less cones, and they're going to really help us with the sense of color, our central vision, and fine detail. And we have really 3 different kinds of cones. We have ones that are going to contain red, green, and blue pigments. And here's a nice little table that kind of summarizes the structures of the eye and their functions. So feel free to read through this to kind of refresh your memory. Now, as far as abnormalities of the eye, some people, mostly males, will have color blindness. And this is a genetic abnormality in which our colors, which typically are red and green, can't be distinguished. So they do a test where they show you a bunch of these little dotted circles, and we'll have a number in there, and they'll ask you what number you find in the circle. For those that are colorblind, you won't see any number at all in here. But most of us who do have our color vision will see the number 27. And the reason we find this more in males is because this genetic abnormality is found on the X chromosome. So females have the XX sex chromosomes, whereas males have the XY. So in females, we replace this abnormality by the good genetic information for not having color blindness by having two X's, whereas in the male, because they only have that one X, they're left with the abnormality. They don't have another X sex chromosome to correct this blindness. I also want to mention cataracts, and this is when we're going to have a cloudiness of the lens. And a lot of elderly patients right around the age of 65, 70 will go in and they'll replace this lens with an artificial one. Also, we have glaucoma. I'm sure many of you have heard of it already. What this is we have pressure buildup within that aqueous humor in the front of the eye. And so from that increase in fluid, we're going to start to feel pressure against the retina, specifically to our optic nerve, where that information is sent over to our brain. And so this can be a huge issue. Usually we can clear this up and pull some of the fluid out of the eye in order to relieve some of the pressure here. And this is the view that we would have because of that. This is normal vision. We would only have nice central vision here, whereas pressure being put on our mostly our rods here, that would decrease our peripheral vision. Okay, next we have astigmatism, and this is a abnormal curvature of the cornea. So you can see here someone that has astigmatism, their cornea is a little bit more pointed than a normal cornea here, and this causes a change of the light ray that's entering the eye, hence a fuzzy image that will be perceived. Nearsightedness is also called a myopic eye, and this is when our eyeball is too long. So what happens is that focus area that's supposed to land on the retina lands short, and so the proper image isn't being projected on the retina, and so we see something as being fuzzy. This could be corrected by a lens, a concave lens, and our focal point where we want our visual rays to kind of all merge together is located on the retina now. If you are farsightedness, this is also called a hyperopic eye, and this is when we have all those light rays come together too far. So the eyeball is too short and we miss our target, which is the retina here. So we can correct this by putting in a convex lens and moving the focal point to the retina again. All right, now we're going to move into the ear. Feel free to take a break and review information on the eye before moving on to the ear. You want to make sure you master the eye before you jump into the ear. So, just like how our eye was divided into 3 different layers, our ear has 3 different divisions as well. We have our outer ear, which is focusing on the purple here, which is going to function in hearing, and this is filled with air. Our middle ear is going to function in hearing as well, and this is also filled with air. And our inner ear is going to function in hearing and balance, and this is filled with fluid. Now let's talk about the outer ear first. We have the pinna or the auricle. So this is going to be the flap portion of the ear, which is shaped like a funnel so that the sound waves can be caught and transferred down into your external acoustic meatus or external acoustic canal, or this is also known as your auditory canal. And this helps move all the sound waves over to your tympanic membrane. And this tympanic membrane is going to be filled with fine hairs and modified sweat glands called ceruminous glands that'll create earwax or cerumen. Now we have our middle ear, and our middle ear will consist of our tympanic membrane. Of course, this is the inner portion in here, and this catches those sound waves and transmits them over to our bones through a vibration. So these bones are all going to vibrate. let's talk about what these bones are. They're the smallest bones in our body. So we have the malleus, which is also known as the hammer, the incus, which is also known as the anvil, and lastly, the stirrup, which is also known as the stapes. Okay, and these will help to amplify the sound. These will all catch the vibration of the tympanic membrane, which will hit against our vestibule and carry that information to our inner ear here. The other thing I want to mention in our middle ear is we have this tube that we mentioned in our respiratory lecture as well as our digestive lecture. This is called the auditory tube or eustachian tube or pharyngotympanic tube. So it has 3 different names. And this tube connects the throat, our pharynx, to the middle ear where these 3 bones lie. And it's used to equalize pressure. Remember I mentioned examples like going up to the signature lounge on the 96th floor. Okay, going up in that elevator causes your ears to kind of pop, so we need to swallow to equalize the pressure within our head. Or when you're on a plane, you know, moving up from the ground up into the air. And equalizing this pressure is important so that our eardrum, the tympanic membrane, does not burst. Oh, I forgot I had these highlighted. Okay, and that brings us to the inner ear. And like we said, our outer and middle ear are going to help with the sense of hearing, but our inner ear will help with hearing and balance. So we have 3 different areas here. We have the cochlea, which is this snail shell shaped area. Okay, that's our cochlea. We have our semicircular canals, and we have 3 canals, one there, and another one here. And these are broken into anterior, posterior, and lateral canals. And we have our vestibule, which really helps to connect our semicircular canals over to the cochlea. So we mentioned earlier that our stapes, which is the innermost bone in the middle ear, this is going to connect over and send vibrations to our inner ear. And it does this through this area known as the oval window. And that causes the fluid within the cochlea to actually, it transmits the sound waves, the vibrations, into fluid waves. So it's just the same vibrations, it's just now moving within fluid instead of through the bones and air. Okay, and our vestibule, let me highlight that there, okay? This is broken up into a utricle and a saccule. Here is our utricle, here is our saccule. And this is going to help us with gravitational equilibrium. So for instance, if you're going up and down in an elevator or you're moving in a car, it helps with information like that. And then your semicircular canals that we mentioned earlier, these are going to help with rotational equilibrium. So if you are sitting in a Ferris wheel, or if you're at Disneyland in those teacups spinning around and around in circles, your semicircular canals are at work. Or even just turning your head side to side. Okay, so within your cochlea, let's just go back to our picture so we can get this in our mind. Okay, here's our cochlea. Okay, within our cochlea, we have this special organ, and it's called the spiral organ, or organ of Corti. And this is going to contain hairs that'll help us with our hearing. The bending of these hairs, okay, so here's some outer hairs. We also have some other stereocilia and inner hair cells. Okay, we're just going to group them together for now. These hair cells are going to bend back and forth, and that's going to cause vibrations that'll initiate a nerve impulse. So see how these hair cells are all connected over to this cochlear nerve. Okay, it's another cranial nerve, cranial nerve number 8, that is receiving information that will go to the brain in the auditory cortex to interpret this information. Now our pitch is going to be determined by varying wave frequencies that are detected by different parts of the spiral organ, and volume is going to be determined by the amplitude of sound waves. So I do have a video for you on the organ of Corti. I'm going to post this in Blackboard so that you can click the link and watch the video, because I think it is a little bit confusing how this works or where it's exactly located in the cochlea. So make sure to watch this video. It really helps to clarify the location. And this is just an image taken from your textbook. We see the cochlea here. And like I mentioned, it's a little confusing to understand where the organ of Corti is. So what your book has done to illustrate this is it's taken the cochlea and we've sliced it in half. Okay, so this little box, we've sliced it in half, and the canal in here that you could imagine is broken into 3 different segments. And so this organ of Corti is found in that middle segment. Here you could see it's zoomed in. Got something that popped up, sorry about that. Okay, here we go. So here you can see those hair cells, and on the very top of the hair cells we have stereocilia. Now when the information through that fluid comes in, it moves something called the tectorial membrane. That causes the hair cells to shift and move, and that information is then picked up by our sensory nerve, which is the cochlear nerve in this case, and that sends it over to the brain. And we can also see this in an SEM here, I believe. Those stereocilia are in the yellow, and that's connected to the hair cells in the purple here. And this is a closer view of our organ of Corti. Again, here's the tectorial membrane that is shifting, causing the stereocilia to move, connected to the hair cells. Information is transmitted along this sensory nerve, which is the cochlear nerve or cranial nerve number 8. Okay, so noise pollution. Most of us have been around some type of noise pollution, whether it's in a loud concert or a loud boom or fireworks on 4th of July, within a nightclub, you know, on a farm or chainsaw. Any of those counts as noise pollution. So that's going to be any loud noise that's over 85 decibels or chronic noise that can damage inner ear cells. And we have environmental noise that can cause mental health issues such as inability to concentrate, an increase in irritability and anxiety. Basically, I'm describing everybody that lives in downtown Chicago. Just kidding. And then we have noise that can cause a loss of sleep and productivity and can lead to even more anxiety. So what should be done about noise pollution? I want to get your thoughts on this, what you can do. And table 15.3 in your book gives you some noises that are going to affect hearing. So they're all listed here. I won't go through them, but it gives you examples of the sound levels that can be sensed when you're in these situations. And then the effect of that. So for example, when you're in a nightclub or you hear a thunderclap, you have hearing loss that is likely. So sometimes, like I said, when you go to a loud concert or you're at a nightclub and they have that loud booming music, the next day or when you leave that club, you might even hear like a muffliness within your ear, and that's because those ear hairs are a little bit damaged. Now in our inner ear, we have those semicircular canals and the vestibule. So I just wanted to talk about rotational and gravitational equilibrium a little bit. We mentioned that our semicircular canals helps with rotational equilibrium. So this depends on hair cells at the base of each of the semicircular canals, or the ampulla. Okay, so here is each of our ampullas. Okay, we see 1, 2, and 3 connected to each of these semicircular canals. So that's, again, the hair cells in those areas are going to detect this rotational equilibrium. So turning your head from side to side, and we can see this ampulla moving with the fluid moving, which the fluid is called endolymph within the ear. And all this information, so when this cupula portion here is shifting, then it's picked up by nerve fibers, a sensory nerve once again. This time it's a vestibular nerve instead of the cochlear. Cochlear nerve will help with the sense of hearing. vestibular nerve helps with the sense of equilibrium. And then we have our gravitational equilibrium. This has to do more with the vestibule. And remember, the vestibule is broken down into a utricle and a saccule. And within this area, we have a membrane that, once again, will help the hair cells shift. And that happens when we have changes in gravity. So, for example, tilting your head forward, that'll cause the otolithic membrane to shift, and that'll be picked up by a sensory nerve again. And this will be also another vestibule nerve. And that is the end of our Chapter 15 lecture. If you have any questions, please feel free to contact me at any time. I also want to remind you real quick that we have our field trip on Thursday at Rush University's Cadaver Lab. I'm going to meet you guys at 600 South Paulina. It's the Armor Academic Building at Rush University, just right outside the bookstore entrance. And then we'll head up to the Cadaver Lab so that we can look at some muscles, and I'll show you some other organs in there as well. Make sure to bring that packet I passed out on Tuesday of last week.